Okay, hello everyone. In the interest of time, let's get started on this next session. The PODS Showcase. Each year, MIDAS awards a series of pilot study grants for research in data science and artificial intelligence, known as the PODS grants, or Propelling Original Data Science. Teams at Michigan receive funding through these awards to support a wide range of exciting projects with data science and AI as the common threads. And as you can see from the teams that are presenting here today, we have lots of exciting research going on through this program. I wanted to mention, please look out for announcement of the next award cycle coming out in the spring. And other than that, I just want to turn it over to our awardees from this year to tell us about their research. And we are starting out with Kim. Oh, and please hold questions to the lunch after this event, since we're moving fast today. Thank you.
Thank you so much. Hello, everyone. I'm glad that I'm the first, and so I will set the example to finish this as soon as possible. So my research idea is to disambiguate names of female scholars, and I will briefly explain the motivation. After this introduction to my lab, I created a lab dedicated to development of technology for equitable entity disambiguation and linkage, and I successfully hired brilliant students across the campus. So here's the main motivation. So there's a famous narrative in science of science called gender productivity gap. That means female scholars are less productive and less impactful than male scholars. And those studies presented this idea, citing kind of the findings from a bibliographic data, where they count the number of publications and also count of citations. But most of them rely on small-scale case studies in very specialized areas. So there's a sampling issue. But another more serious issue is that they don't properly resolve the name ambiguity of female scholars. For example, not female scholars, but if you type my name on Google Scholar, you will see that I have published more than 300 papers and attract more than 10,000 citations. So I'll be a full professor and renowned scholar at University of Michigan, but that is not true, unfortunately for me. So that is name ambiguity problem. But the female scholars' names are ambiguous in a little different way, because many of female scholars change their maiden names after marriage. So that means they have published papers in their maiden names in certain publications before marriage, and then after that, they might have used the marital names changed after marriage. So previous studies do not consider this possibility of name change and its impact on the counting of the production and citation. So here I started my research hypothesizing that the gender productivity gap studies might be flawed because they are heavily relied on the bibliographic data, where female scholars' names are not properly distinguished. So here's three goals I aim to accomplish using this PODS grant. The first one is to create large-scale label data to train algorithmic models to merge the same author entities split under different names. And this model can be applied to other name change cases of someone who has changed their name after religious conversion, as well as the female name change. And the aim two is to implement the entity disambiguation models learned from the aim one on author names recorded in PubMed data that indexing almost 100 million scholar names in biomedical research. And then I'll try to find out whether these algorithmic models for name disambiguation can successfully distinguish the name change cases of female scholars. This is very important because in biomedicine, it's estimated that 50% of the authors are female scholars. The final goal is to analyze the disambiguated PubMed data from AIM2 to demonstrate how the correct identification of female scholars can actually provide us a different understanding of research productivity and impact of scholars in biomedicine. It's a high risk stake. So if after we disambiguate all these name ambiguity and then still find the gender gap narrative, productivity gap narrative, and there might be a bad news, or maybe we can have a more the accurate representation of the scientific productivity and impact of female scholars compared to male scholars. So, so far I have worked with students to clean and label data of scholar profiles. In computer science, 5,000 scholars in top US universities, and mostly male-dominant field. And also I gathered data from the scholar profiles in nursing schools, almost 1,500 female-dominant area. And also I pick up the communication area because there's kind of a balance between male and female scholars. And this is the most time-consuming and laborious part, and students are working on it. And meanwhile, I also passed the entire PubMed baseline, and I'm cleaning it for disambiguation. And I found that there are more than 70 million paper records with about 500 million name instances after 2000. So I'm currently rewriting code for machine learning procedures developed for another NSF project. I was the principal investigator. So I think that we'll get the preliminary results soon. So I hope I can share a more exciting news from this PODS grant project. Thank you so much.
Good morning. My name is Will Weaver. I'm a PhD candidate in the Department of Ecology and Evolutionary Biology. And today I'll be talking about one of our ongoing projects, which is using machine learning methods and computer vision to extract trait and morphological data from digitized herbarium vouchers. So just as a quick overview, an herbarium is pretty much just a library for dried and preserved plant specimens. For several hundred years, botanists around the world go out into nature, take a specimen, and then preserve it for future research. And up until about 30 years ago, if you wanted to use these specimens in your research, you probably would have had to go in person to an herbarium to physically hold the specimen. But recently, most of the herbaria have begun digitizing their collections. So now we have data sets of hundreds of millions of images of plant specimens. And obviously that represents limitless potential in botanical research that we can do with those specimens. And one of those projects that we're working on is that we're interested in understanding the emergence of complex features in flowering plants. And there are about 350,000 species of flowering plants in the world, and we can only do so much with genetic and transcriptomic data. So we're looking to incorporate morphology at scale to increase the usefulness of our phylogenetic analyses. So for our machine learning components, this is the general architecture that we're trying to follow. Each of the green boxes is some kind of machine learning component. We're trying to analyze the specimen first by breaking it down into components and then conducting component-specific analyses. One of our biggest focuses is efficiency. We have to generate a lot of our own training data, so if we can scale down the scope of the required training data, that's great. One of the first steps in this process is first defining the archival components and then the plant components. For archival components, we're interested in transcribing label data and also locating the rulers so that we can convert pixel distances into metric distances. And then for plant components, At this stage, we're interested in filtering out usable pieces of the image that we can actually get measurements from. So if you can see the image on the right, there's a leaf that is unusable because it's hidden, so we're not going to try to get any data from it. Once we've filtered out our data so that we have smaller images to work with, then we can do more computationally intensive machine learning architectures such as instance segmentation to generate masks or landmark detection, in our case, to find different morphological measurements on each of the leaves that we're interested in. And as far as broader impacts, this project will generate a very large data set of plant traits. And to date, it will be one of the largest projects to date using herbarium specimens. And one of our goals is to create a database, an extended specimen database, so we have data derived from the image that is tied to a specific herbarium specimen. And our machine learning ensemble is designed to be customizable so that other researchers can easily take modular components and add on their own so that they can detect new traits. We're also working with some of our collaborators to directly integrate this software into data aggregation portals so that our data analysis can take place at the time of deposition into a data portal rather than doing that after the fact. We're also working on a project called Field Prism to take pieces of this project out into the field so that we can create higher quality archival imaging when we're in the field. Oftentimes, when plants are dried, the color changes, the size changes, so it's really useful to have a before and after image. So we're trying to take that out into the field at the same time. Yep, and I just want to thank our lab and all of our collaborators. Thanks.
Hello, my name is Dojun. Thank you for joining us today. Our project is developing language-based tools for real-time counseling feedback. I'm a PhD student at the Language and Information Technologies Lab and also the presenter for you today. And our team includes our principal investigator, Veronica, who couldn't be here today, and our co-PIs, Radha and Kenneth. Let's begin. So here's some background. In 2019 alone, 24% of American adults with mental health issues reported unmet treatment needs. And there is a current shortage of mental health workers. As you can see on the left of the figure, the number of people being referred for mental health treatment has increased. And you can see on the right, at the same time, the number of people waiting for that treatment has also increased. So this is not good in that people who are in need of mental health care are not able to meet their needs. And there are several important barriers to the rapid training of new counselors, which can actually mitigate this problem. And some of these include the need for extensive clinical supervision to train these new counselors, and this supervision entails the laborious process, and a lot of the times it's very expensive to do this training. So in this project, our goal is to use NLP to tackle this problem. Our first goal is to generate new computational methods to provide language feedback to health professionals while they learn counseling skills. And we believe that this will allow us to provide cost-effective training resources for the development and maintenance of counseling competence. So let's take a look at our proposed method. Our method consists of three major aims. Our first aim is to build a data set of counseling reflections. And we can do this by starting with a seed set of client prompts, which expresses common concerns or behavioral topics that arises in counseling domains. And we can, with the help of motivation interviewing and other counseling experts, we can annotate these prompts with varying levels of reflections or responses that counselors might give in response to these prompts. At the same time, we can also utilize transcribed conversational counseling sessions between counselors and care providers to augment our data set with client reflection pairs, which we can then use our data augmentation method to create a data set. After we create a data set, we move on to our second aim, which is to develop computational methods to score and classify reflection quality. Now, given that we have a data set with a prompt and varying levels of reflection qualities or response qualities, we can now utilize computational methods or models, for instance, a label classification model or multi-level margin ranking approaches to derive this, to turn this data set into a model that we can use to derive a model that can derive qualities or levels of the reflections. And finally, we also plan to utilize this data set and the scoring model to build a reflection writing system that can take in user-written responses, counselor responses, and produce a rewritten version of the responses that expresses a higher quality of reflection or counselor behavioral strategy. So our plan is to do this create this rewriting component with the help of fill in the gaps approach with reinforcement learning. For instance, that can be done with denoising autoencoders. We can also explore text-style transfer approaches to turn the style or the semantic meaning of this user-written response to have more empathetic or reflective qualities. So far, we have completed smaller scale experiments of AIM-1 and AIM-2, and we hope to scale these approaches and experiments with a more larger data set and in the wild collections. And finally, we will also work on the AIM-3, where we actually build the reflection or counselor response rewriting system and deploy it in a user study. We believe that our work will have a broader impact in the sense that the proposed research can provide the necessary counselor training and coaching, both at the beginning of their career as well as for continuous learning throughout their practice years. And we also believe that the feedback tools could be deployed in educational settings and provide a supportive learning environment that allows students to make mistakes and learn from them at their own pace. Thank you.
Hello, everyone. I'm so excited to be here to present our work of building a genomic literature knowledge graph. So if you are a biomedical researcher, you spend a lot of time managing literature, searching for data sets. So this is current what we are facing right now. So for addressing one question, like what's the relationship between the two genes, people will look for the literature. Usually you will look up the literature from PubMed or Google Scholar. And then on the other side, you will look for some genomic database, like Ensemble and many other databases. So right now, the problem is the two sources of knowledge are isolated. So you have to like jumping between the literature and databases, and then you are the human is the one who kind of merge the knowledge together and perform all this research afterward. And in the end, when you publish your work, you still publish on PubMed and then deposit your data set into the databases. So there's a huge problem kind of between the gap between the literature and the databases. This also reflects the unstructured knowledge in the literature and structured database in these databases. So therefore, you can see with this gap in between, we cannot enable any this AI-based guided tools to help you to relieve this work from literature search and knowledge understanding. Therefore, to tackle this problem, we propose, I think there's a really ambitious project to build this genomic literature knowledge graph. So this is actually a part of this knowledge graph in our database. So it's kind of already combine the literature and the database in this way. So for this part, this node in green are the entities in these genomic databases. It's kind of curated from many large-scale consortium like ENCODE, GTEx, or many people put a lot of effort build this data set, and we put them together, already build a knowledge graph for this from these databases. And on the peripheral, we have 3 papers showed here, and we are able to extract all these genomic entities and relationship mentioned in the literature. So in this way, we are able to connect these 3 literature papers with these structured databases by using these terms. So in this way, we are able to merge like 30 million PubMed literature with all these large-scale consortium databases like this. Kind of out of battery. So this is the part how we consolidate these genomic databases. So the thing we really focus is leveraging a lot of ontology in the domain. So we cannot just build a knowledge graph out of nowhere. We have to use all the identifiers in the literature and people usually use it. So the hope is once there's other people built a knowledge graph, we are able to merge. So this is really the key. So right now we already identify 600,000 genomic terms and 1.8 million relationship among them by consolidating many consortium data like this. And then with this 30 million literature from PubMed, we use many tools from NLP like name entity recognition, relationship extraction. So by doing this, we are able to kind of giving, on the top, this is abstract from the literature. We are able to use machine learning tools to identify the entities and relationships, and then map to this green entities from the genomic knowledge graph. So in this way, we already kind of process 33 million articles, and then we have 188 million genomic mentions and 80 million genomic events from the literature. So with this knowledge graph built, we can see we can empower the literature review, like retrieval. So when you query the gene, like TP53 regulate another gene, MYC gene. So traditional, if we use Google Scholar or PubMed, they just only match the keywords. But in our knowledge graph, we are able to match this pattern, these two genes regulate with each other. So therefore, the literature like retrieval will turn a pattern match in our knowledge graph. And another, I think, also very exciting result is we are able to kind of summarizing all these relations among the, all these genomic terms. And then we are able to tell more, like with more evidence, like which of the gene relevant to the tumor, but by looking at, using the mouse. So these two terms, TP53 and this neoplasma mentioned like over 76,000 times in the literature. Therefore, I will rely on this and give the result. This gene is more related to tumor. So another exciting application of this is we are able to summarize the field. If you given the query, we will return like hundreds or thousands of papers, and then just with our knowledge graph, we are able to summarize the knowledge to you so that you don't have to track all this literature. So we are right now setting up the web portal, and we are improving the information extraction part, and we are also starting doing this hypothesis generation so that biological medical researcher can generate the hypothesis automatically from our knowledge graph. And in the end, we are also able to kind of identify the contradictory knowledge from literature. It's that. Thank you, everyone.
Hello, everyone. My name is Maggie Reuter, and I'm a postdoc in the biomedical engineering department, and I'll be presenting on behalf of our team. And I'll be talking about combating antibiotic resistance using machine learning. So to jump right in with some background, pathogens are becoming more drug resistant to our antibiotics. And domestically, you might recognize some of these bacteria. Staph and Pseudomonas, among others, are costing our healthcare system billions of dollars every year. If we look more globally, Mycobacterium tuberculosis actually caused 10 million infections in 2020 alone. And so the way that we're currently treating these drug-resistant bacteria is to use combinations of drugs instead of a single antibiotic. And so in the case of TB, this is actually a combination of 4 drugs over the course of 6 months. These current methods were actually based on empirical data, and they haven't been updated in decades, especially for the TB treatments. And so our group is actually trying to develop methods to better predict how to use these drugs in combination, basically seeing if 2 drugs will work synergistically with each other or not. And so the question then becomes, if you have thousands of FDA-approved drugs, how do we choose effective combinations without having to do millions of experiments? And so our current predictive models actually use omics data. And this can be costly experimental data, but currently this is sort of the state of the art for predicting drug-drug synergy in a computational way. And so we can actually simplify this omics data into sort of a interaction between a drug and a gene or a drug and a metabolite. And here I have just a binary matrix where a 1 would be a strong interaction between a drug and a metabolite or a drug and a gene, and a 0 is sort of a weaker interaction. And we can use that to actually predict drug-drug synergy. Our group is actually trying to use drug-protein interactions in place of these drug-gene interactions or drug-metabolite interactions. And the benefit of that is that we can use computational methods to do that. And so in our case, we're actually using 2 different computational methods, deep learning and molecular docking. Molecular docking is kind of the standard interaction between a ligand and a protein, and so we're using that as a basis, and then we're trying these new deep learning methods that are advancing because of stuff like AlphaFold that's new and things like that. So the deep learning methods are sort of the new exciting part of that aspect. And so again, we can just get a sort of simplified, very simplified binary matrix, and then use that in a machine learning model to predict these drug-drug interactions. And so there's actually 2 aspects of our project that we've been working on, the first being this machine learning model. And I'll just show some brief results. This is just like an accuracy metric for a couple different algorithms that we're using, and then our 5 different data sets, the blue and the red being the computational data, and the orange, gray, and green being that omics data that we originally had. The second aspect of our project is to actually use all of this data that we have, both the omics and the computational data, to try and learn some drug mechanisms of action or biological principles behind synergy, things like that. And the way that we're doing that is to create big heat maps of these binary matrices and try to connect them back to higher level functions, metabolic pathways, things like that. And then we can also do sort of even simpler statistics to just compare the data sets and see how similar they are. So I just have an example of that here. The darker color means that the 2 data sets are more similar to each other. And then just some key takeaways that we have. Our computational data is actually predicting drug synergy and antagonism just as well as chemogenomics, which is kind of like our standard that we're trying to beat. And this is really cool because then we might be able to use these models to predict drug synergy for emergent diseases or stuff that you can't model experimentally. The second aspect of this is we actually were able to see some patterns in synergy for rifampicin in particular. I'm not going to go into too many details here, but I will be at lunch, and so if you're interested, please come talk to me. And then I just wanted to show some broader impacts that we have if we are able to replace this omics data with drug-protein interactions that are computationally predicted. We can create a kind of pipeline for creating, like I said earlier, like models for emergent disease, or even just the bacteria I mentioned in the beginning. And our group is actually working right now on creating a model for MTB, so Mycobacterium tuberculosis. We can also potentially apply this model for other small molecules that might interact with drugs in our system, so heavy metal stressors or oxidative stressors, natural products, metabolites. And then we're continuing to try to make this model mechanistic and explainable to non-AI experts, more biological-based explanations, and those drug modes of action that I talked about earlier. Cool. Thanks.
Good noon, everyone. My name is Tara Ansari. I'm presenting this project. The PI is Dr. Negar Farzaneh, but she wasn't able to attend, so I'm presenting for her. And this is the work that's being done at the Weill Institute for Critical Care. So the work is focused on improving classification of cardiovascular diseases using ECG, but incorporating hierarchical relationship between different arrhythmias and the uncertainty that exists in the labels that we use. As you probably know, cardiovascular disease is the #1 cause of mortality and morbidity around the world and the US. And the the most common diagnostic tool for that is electrocardiogram. And there are tons and tons of papers published on using deep learning and rule-based approaches, you name it, for doing classification of ECGs. But that's not what we're focusing on. We're not focusing on building a better model. We are focusing on a better model, but not in terms of like the model architecture, but in terms of like how we define the problem. The most important issue is that when you actually try to use these models clinically, the way they're trained, some of the predictions don't make sense because they don't take into account the hierarchical relationship between labels. And also they don't take into account the uncertainty. So our hypothesis is that if we incorporate human domain knowledge about the relationship between labels, we would end up with a better model. So this is an example of what I'm talking about. So this is called SNOMED CT hierarchy. It's a system that has all the diagnoses and medical concepts. And so you see here, you see here that supraventricular tachycardia and ventricular tachycardia are both children of tachycardia. But the way all models that I'm aware of treat this problem is to consider tachycardia and supraventricular tachycardia and ventricular tachycardia as independent labels and train a model. So everything basically is an independent label. So you may end up with a prediction that an ECG is supraventricular tachycardia, but it's not tachycardia, which is impossible. Another issue is like here when you have a node that is the children of two other nodes. Again, the same problem exists. Some of these, the children of some of these nodes are mutually exclusive. So, for example, we don't show this here, but ectopic beat, the children of that are PAC, premature ventricular contraction, atrial contraction, and junctional contraction. Those are mutually exclusive. You can't have them at the same time. But that's completely ignored in the way the current models work. So our solution is to use a hierarchical classification approach, but also the hierarchical classification approaches that exist are relatively rudimentary in the sense that you basically train a model to do the first level classification, then whatever you get, you go to the second level, you do another classification. You basically train a classifier for every level of your tree. And they require that you have all your labels all the way to the leaves, which we don't have. So these are the labels that we have. We're using a data set of 2 million ECGs that have been done as part of clinical practice at U of M. And when a clinician reviews these ECGs, they may call it tachycardia, or they may call it supraventricular tachycardia. So how far they go into the tree is not consistent. So you may get this label, you may get this label. So we're trying to basically use all of those labels to the extent that they give us information. So we're approaching this two ways. So two approaches for this. One is to model the conditional probability of each of these nodes conditioned on its parent. That's a more, I would say, closer to the traditional approach. It has some benefits, some drawbacks. The other approach is to train a function on each of these nodes and do a post-processing step that would allow you to, from those outputs, to calculate the conditional probability and then put that directly into the loss function. And so that would allow you to do training on the entire tree in one shot. So we're going to compare those. The second aim is modeling uncertainty. So you get labels that say maybe or possibly, probable this. So there are a lot of uncertainty. This is more of a clinical aim in the sense that we're only focusing on left ventricle hypertrophy, LVH, which is the one label that has the most amount of uncertainty about it. We have 211,000 of those, and around a fourth of those are uncertain. So the way we would address that is by using soft labeling, which considers the certain cases of positive and negative, but also assumes a probability for the ones that are uncertain. And the way we're going to calculate this probability is so for a subset of these patients who have ECGs, they also have echocardiogram which would give you the true gold standard for whether they had LVH or not. So we extract those from the electronic health records and then see what percentage of those actually had LVH, and that would give us P, and then we can use soft labeling for that. So in short, in addition to the clinical broader impact of doing better classification of arrhythmias, we also think that the new models that we're building would enable other problems that also have the hierarchical relationship in their class structure to be done more effectively. Thank you.
I'm Joyce Penner, and we are a team from climate and space sciences and engineering and statistics, and a couple of students who are here with me, as well as Young from Department of Statistics, students from computational sciences. So the problem that we are trying to understand is what is the forcing of climate by aerosols. And I'm trying to illustrate two kinds of forcing here. Direct forcing on the left. That's where when you introduce more particles, more aerosols into the atmosphere, you have more reflection of solar radiation, so you have more cooling. The other forcing is more indirect forcing. That is, when you add aerosol particles in an updraft that forms a cloud, the number concentration of droplets increases, and that reflects more solar radiation and further cools the climate. Only observations by themselves are unable to be used for estimates of forcing. So in the climate community, models are used, and the differences in models are used to estimate the uncertainty. Some models, another issue is that some models fit some observations while other models fit other observations, so it's very hard to say this is the best answer, and instead we just take the average. What we plan to do is to use machine learning to sort out which processes cause differences between different models. And then we also want to find out which processes cause differences between models and observed aerosol optical depth to figure out what processes need to be changed. And when we determine those processes, we hope to be able to use them to correct the processes, or at least agree on the best way of determining processes, and thereby hopefully lead to smaller divergences between predictions of models. So I have an aerosol model that I have been developing with grad students over the years. It has been used to provide the features, that is, all of the processes that go into predicting aerosols, and also using to predict the output of the machine learning method for AOD. And we are currently using, two students are using two different machine learning methods, convolutional neural networks and gradient tree boosting. And we are using Shapley values to determine which of those features or input are most important for predicting AOD. And the next step will be to substitute, instead of the chemical model AOD, the AOD measured by satellites. And that way, hopefully I can figure out why my model doesn't always fit the observations, or at least which processes are important for that. And then I'm going to find a second aerosol model, and we will use that to do another machine learning, and then we will swap the feature inputs from Model B into Model A to figure out what is, and we'll go through that for each of the features to try to understand what it is that is really different between the two models. And that's all towards getting at that first indirect effect, and then we'll be doing that again, going through those same steps to try to predict the droplet number concentration. The broader impacts are that, you know, at the present time, there's something like 30 models out there trying to predict AOD and droplet numbers. So we will, if we figure out how to do this with our two models, we will go on to make a proposal, and hopefully engage all of the 30 models. And this may be hopefully able to lead to a convergence of how to treat different processes and a convergence of estimating forcing by those models to lower that uncertainty. And that's all. Thank you.
Hi, I'm Amy Gordon. I'm an assistant professor in social psychology. I primarily study close relationships and well-being. And I'm excited to be here today to talk with you very briefly about the work I'm doing with a number of wonderful colleagues to develop a large-scale data set to track romantic relationship formation and maintenance. So to put this work into context, I just want to start with a few numbers. So in the past couple years, each year, the revenue for US online dating sites has been over 600 million. There are over 44 million US adults who are currently online dating users, and over half of them are smartphone app users who primarily use dating apps on their phones. Now, bringing this closer to home, last year, over 12,000 students at the University of Michigan participated in something called the Michigan Marriage Pact. So I'm not sure how many of you have heard of this, but I found out from my own students. Students complete a battery of questionnaires, and then at the end of that, they are sent an email with one match to another student who participated as well. So again, 12,000 students participated in this last year, very minimal advertising. And so these numbers inspired us to think about developing a dating app for research purposes. So we're in development right now. We're working with the amazing LSA Tech Services team and developing both the front end user experience as well as the back end, accessing and storing the data securely. And the goals for this project are twofold. So the primary goal is to track a large group of individuals through mate selection, relationship formation, and relationship maintenance. So in order to understand compatibility in relationships, we really need to know who people are when they enter into relationships, as well as who they become in their relationship, through their relationship experiences. Historically, that's been very difficult to do at the large scale that we would need. And the advances in computational modeling have only recently made this something that we could think about doing at this large scale, along with this sort of popularity of dating apps. The second goal is to actually generate a data set for developing methods to preserve privacy of sensitive data. So as we move towards an online and more transparent science, we're putting more of our data sets out there. So as a relationships researcher, I can tell you a lot of that data is highly sensitive. And figuring out how we can do that safely and protect the privacy of our participants is really important. So we hope this data set will be used by computer scientists and engineers for this purpose. So I'm not going to go into each of the details of these methods, but to highlight a few key points. So one is that this app is being built specifically for students, separate pools for graduate and undergraduate students, but they will have to verify their student status each time they sign in to maintain sort of this select pool for safety and security reasons. Initially, our goal is minimal interference. We're trying to track people's experiences rather than change them. But over time, we hope that the information that we gain might actually help us develop algorithms to better predict compatibility and to see if we can actually run experiments at the initial dating phase and look at how that changes relationships over time. Within the app, in addition to collecting all of the information about people's experiences with matching with partners, we also are building an in-app messaging, which means that we will have all of the text information from partners as they first meet up in the app and talk to each other. And so the opportunities for this rich text data, I'm very excited about. Finally, in addition to sort of the typical experience you might see in a commercial app, we're also building in in-app surveys, not just the background surveys that tell us who they are, but also surveys throughout their experience of the app in order to capture information about why they make the choices that they do, as well as information about their dating experiences. And then finally, for participants who let us know that they've actually established a relationship with someone, we'll be inviting them to join a separate study outside of the dating app so that we can continue to track them over time in a way that hasn't been done before. So there are many broader impacts of this work, both in terms of thinking about it from a relationship science and data science perspective. But it's not too exaggerated to say that the quality of our relationships determine who lives and who dies. And so I hope that this data set will help us to bring together relationship science and data science in order to answer questions we haven't been able to answer before about what promotes a high-quality relationship. And hopefully that will help us shed some light on this larger question of well-being. Thank you.
We had one team that wasn't able to join us today, so.
Hi everyone, my name is Qing. I'm an assistant professor in ECS department. Today it's my great pleasure to give you an overview of our work on this machine learning guided co-design for reconstructive spectroscopy. And this is a joint work with my colleague Pei Cheng-Ku and his group. So a spectrometer is a device for measuring wavelengths of light over a wide range of electromagnetic spectrum, and widely used for analyzing sample materials in chemistry, life science, and biomedical sensing, etc. And conventionally, optical spectrometer is something like this, that you have a sample, you inject some excitations, and then because you want to measure the spectrum of the sample, you use some dispersive or diffractive optical elements to disperse the spectrum to different directions, and use this photon detector to detect different wavelengths of the light. However, this conventional optical spectrometer is quite expensive and also quite bulky, mainly because the optical instrument that you are using. But for many applications that you want to design some cheaper, smaller spectrometers, for example, for this epidermic sensing device for health monitoring, you want to design a spectrometer that's really small that you can stick it to your skin. So the goal of this project is that we want to design a cheap scale spectrometer by eliminating the optical components. And this can be achieved by local strain engineering, which is leading by my colleague Pei Cheng-Ku. And another key component of this cheap scale design is by using optimization and machine learning approach for spectrum reconstruction. So I'm not an expert on this cheap design, so this is majorly done by my colleague Pei Cheng-Ku. So the major idea here is that we want to design some chip with selective frequency cutoff rate for different wavelengths. And by doing this, actually, you can design a cheap scale spectrometer that's really small. This is an illustration of the size of this spectrometer. However, there's a trade-off, right? So if you get off the optical instrument, then you need to solve a reconstruction problem. And mathematically, this problem can be formulated as following, that you have some measurement which is composed of 16 photon detectors, and you have a given library, and your task is that you're given I and R, sensing matrix R, you want to reconstruct the spectrum S from the given measurement and R. And this problem is quite challenging. First of all, usually this R matrix, this sensing matrix R is underdeterminate and sometimes L conditioned. So there could be infinite many solutions to your problem, to this linear system. And also it's highly sensitive to the model mismatch. And in this project, we designed some optimization approach to solve this reconstruction problem. And the idea here is that the spectrum, if you look at the right-hand side figure, is quite smooth, right? So we added certain regularizations to the solution, that we added TV regularization to promote the smoothness of our solution. And also the spectrum is non-negative, right? So we are solving a non-negative, a TV regularized non-negative least squares problem to reconstruct the spectrum. And based on this, actually, we can achieve already pretty good reconstruction result. We can not only like reconstruct the peak of the spectrum, but also we can get a pretty good estimation of the whole spectrum. And this project leads to accurate and robust and efficient reconstruction of a smooth spectrum using actually very few photon detectors. And the broad impact of this work is that actually we can design a cheap scale spectrometer for commercial, many potential applications in chemistry, life science, and health monitoring. As a future work, in this model, we still have some model mismatch between the real data and then the assumption that we make. So to improve this reconstruction, so one is that we want to improve the chip design to better match with our model, have more accurate measurements. And secondly, I want to design deep learning methods for improved efficiency and robustness by using some simulated training data and test on those real data to improve reconstruction. And this project already, we have some published work recently in Nano Letters this October that for this, this protocol of this cheap spectrometer using this optimization approach. Finally, I want to thank all my collaborators and thank MIDAS for this generous support for our project and secure for attendance. Thanks.
Thank you all. That concludes the PODS presentations. Yeah, if you could help me with bringing up the next thing. The next thing is to recognize the people who made the posters that many of you hopefully enjoyed yesterday. Our current MIDAS postdoctoral fellows had a judging committee led by Bernardo Modenesi, and I'm going to invite him to come up and talk about what they did and what they liked.
Thank you, Jag.
You have a clicker here to advance
Thank
slides?
you. Okay, I can use.
Use the keyboard.
So, as Jag introduced me, I'm Bernardo Modenesi. I'm a postdoc at MIDAS, recently joined. We've been having, MIDAS has been having the tradition of awarding posters in the summit since 2015, way before I was here. and I was glad to be the chair of the committee for the poster award this year, together with other postdocs, the Professor Murali Mani and Majid, also another researcher fellow. And we decided this year to give 3 category prizes, specific prizes, and 1 best overall poster. So for the first category prize, I want to invite Natasha Vernoy, PhD student in psychology and co-authors, if any of them are here today, for their promising language-oriented research. They're developing Spanish-English corpus, the biggest Spanish-English corpus, and they're going to make it available to the public, to researchers, to foment research in language, which is quite impressive and really good for academia. So if any of the co-authors are here, we're going to save her her prize, and we're going to hand it to her later. For the second specific category, we have, I want to invite the PhD in physics student, Tai Gao Ma, for the best layout and visualization and clarity of exhibition. They're, Tai Gao here? No? No. Please come. Yeah. So they're using reinforcement learning to design multi-layer films. This has optical and energy applications. So we want you to receive the award. And a little pen from MIDAS. Congratulations. A round of applause for Tai Gao. Thanks. All right, our next specific category award is gonna go, I wanna invite Kais Riani, PhD student in computer science and information sciences, for their innovative use of data studying the human circadian rhythm with a non-invasive procedure. Please, if Kais Riani is here, or any of the co-authors, come to the stage. If not, we can save your prize for another time. All right. So finally, we have the best overall prize, which was hard to choose because we were considering research impact in academia and society. Oh, there you go. You want to receive your award? Okay, Natasha Vernoy. Welcome. Yeah, now I realize that other folks were watching from the other room. Okay. No, no problem. Okay, here you go, your award. Yes, congratulations. And a pen for you. Yes. Thank you so much. Yes, a round of applause. Is there any other author from Kais Riani's paper that is around? No. Okay. So we can continue. So yeah, for the best overall poster for this year. We considered clarity of exhibition, academic impact, societal impact, and many other things. And it was hard to choose, but we decided to give it to Harkirat Aurora, PhD in biomedical engineering. Harkirat, if you're here, please come to receive your prize. It's interesting that Harkirat is using data science for treating drug resistance infections, which is pretty relevant. If none of the authors are here, so then I'm gonna hand the microphone to our director for concluding remarks. Thank you so much.
Thank you, Bernardo. So
Thank you.
that brings us almost to the close. Sorry, go ahead. So I don't know about you, the past day and a half have been very informative for me. I've learned a lot about a broad variety of wonderful work that so many of our colleagues are doing here. I just wanted to take a minute to remind you what MIDAS does. And we have our 3 areas of or 3 ways in which we try to promote data science and AI throughout the university. We support and enable and facilitate research, of course, primarily. But to be able to do that, there are a number of training things that we do. And so if any of these things are appropriate for you, please reach out. Please, we'd love to be able to help. And finally, for getting stuff done, what you've seen today and yesterday is that most of the work requires multiple types of skills and collaborations across disciplines. And so the number one thing that we try to do in terms of just creating an environment in which good work can get done is to help you meet people. And so I hope you've been able to do that in the past couple of days. And please do that over lunch today as well. Before we go, I want to recognize our program committee for designing a wonderful day and a half for all of us. Let's give them a round of applause. And our staff and student assistants, many of them weren't even in the room at the beginning when I introduced some of them, but now I think as we're getting towards the end, at least some are here. If you're in the room, please stand up so that we can be recognized. And as you can see, several are still not here. They're still doing things outside. But thank you very much. You really did a fantastic job, and I hope all of you will thank them if you meet them over lunch. And with that, I believe lunch is set up. So enjoy, and hope to see you very soon.